Hi. So I'm Carlos, and I'm a PhD student at the Foresight Institute, and I'm going to be presenting on this idea that I submitted for the Hypothesis Prize. And the title of the proposal was Harnessing Rejuvenation Events that Occur During Embryogenesis. And just to start on the topic, let me highlight some of the really important biological processes that occur during embryogenesis. So as probably most of us know, there is a lot of tissue, young new tissue being generated during embryogenesis. And all of this is occurring without any type of replicative senescence or large accumulation of molecular damage. And also, embryos are incredibly resistant to cancer. People have done these experiments where they take embryos and they inject them with cancer cells. And as long as this is done before organogenesis occurs, the embryo takes complete control of the cancer cell. It doesn't even kill it. It just stays there, and the animals are born, and you can still track the cancer cells. So I think this ability to control cancer is remarkable. And in addition to that, something that is really important is that thanks to work that has been done by the Galicia lab, now we know that there is a rejuvenation event occurring during embryogenesis that takes epigenetic age back down to 0. And I think some important questions regarding this is whether this is the only rejuvenation event that is occurring during embryogenesis, or if there are more. And I think it's also really important to figure out how exactly the embryo is doing all of this. So the central idea of my proposal is how can we harness these embryonic mechanisms for therapeutic approaches to combat age-related decline. And I think that a good place to start is to have a really deep understanding of everything that is occurring during embryogenesis. And I think that to have that level of deep understanding, we have to start even before embryogenesis occurs, so with the gametes. So I structured this proposal in 3 different phases. And the very first phase is to identify age-related changes that occur in gametes and that the embryo is going to need to rejuvenate. So the way I propose to do this is to take gametes from individuals at different ages and then do very deep phenotyping and characterization of the molecular components of these cells. So these will include things like organelle function, proteomics of the soluble and insoluble fraction of proteins, transcriptomics, metabolomics, lipidomics, as well as covalent changes or covalent alterations to the DNA. So for an example of this is methylation, but there are multiple other types of DNA modifications that occur, and now we have the technology to map them at a genome-wide scale. So I think we should be looking into that. And the final product of this will be a model in which we have all these different trajectories of the molecular features that compose the gametes. And what this will allow us to see is what's changing with age in these cells and what are the trajectories that underlie those changes. And once we know what is changing in the gametes, the next step that I propose is to observe how the embryo rejuvenates those changes. And the way I propose to do this is similar to the first phase, basically taking embryos at different stages of embryonic development and repeating this deep phenotyping and characterization so that then we have a complete model of the age-related changes that occur in the molecular features of the gametes, and then we can see what the embryo is doing with them later on. With this data, we can identify specific events that might show some promising therapeutic potential. So in this example, I labeled this mitochondrial dysfunction, but the idea is to do it in an unbiased manner, right? But let's say that what we're looking for is for changes that have this trajectory in which we see a correlation with gamete aging, and then we see a reversal during embryogenesis. And then the third phase of the project will be to try to copy these events or rejuvenation mechanisms that we have identified and try to apply them to different biological contexts so that we can use them for therapeutic approaches. And there are multiple ways of tackling this problem, but one of the ways I propose is that once we identify this specific rejuvenation mechanism that we're interested in, we can actually, because we already have this data on the model, we can identify peptides, proteins, genes that correlate with this specific event. And from that, obtain the transcriptional signature that correlates with this event. And once we have a transcriptional signature that correlates with this event, we can go and search for small molecules that produce a similar transcriptional signature. And the way one can do that is there are these databases that characterize the transcriptional responses that different small molecules make cells do. So people take cells, then treat them with a small molecule, then measure RNA levels, and they have done this for thousands and thousands of different molecules. So then you can go to one of these databases and you can search for your specific transcriptional signature that you're interested in, and you can find either small molecules or a combination of small molecules that are predicted to produce the same transcriptional signature, use it in different cell models, and see if you can recapitulate these protective mechanisms that you have extracted from the embryo. And like I was saying, there are multiple ways of tackling this problem. This is just one way that I have in mind. But other benefits that can be obtained from this type of deep phenotyping and characterization is that we can perform very complex screens because we are narrowing down the search space. So because we know what genes are correlating with a specific event, we don't have the need to do a genome-wide search for candidate genes. We know exactly which ones, so we can perform more complex screens like combining different genes or doing screens in embryos, which is technically challenging. We can also reconstruct mechanistic networks with different types of computational approaches, so we can have some mechanistic insight into what's going on. And something that I think is going to be really important, it's to determine which of these rejuvenation events actually require cell division to occur. I think that as we try to take the Yamanaka factors and other embryonic-inspired protective mechanisms into other tissues, I think it's important to understand what are the conditions necessary for these rejuvenation events to occur. So that's my presentation. This is my email. And if you have questions, comments, or just want to chat, I'm always happy to chat, so reach out. Thank you.
Thank you so much. We stayed uninspired. This is wonderful. Questions, comments? Okay, we have a few. Colin.
Okay, this is more comments. So
Yep, do. Comments are very welcome here.
So back in the days, people have taken tumor cells and put them in an embryonic extra matrix, and they were able to reprogram the tumor to lose the tumor genesis, right? So I would bet on the extra matrix there. Do you make any inroads to figure out how the interactions you have between the cells in the embryo to influence your reprogramming?
That's great. I actually have read on how, because some people follow up on this, so they kind of map like the pathways that are downstream of this, but I hadn't thought of what's upstream of it, so thank you, that's a great suggestion.
Yeah.
I just want to say that I specifically have a research program working in this direction, and I would like to connect to other people who work or would like to work in this area. I think we should have a working group going.
We will have a working group on this, absolutely. Great, thanks.
Yeah, just a general kind of open-ended question. Because we don't have the benefit of growing our somatic cells to like orders of magnitude larger than what they are to begin with, like how do you uncouple the sort of effects that you might see in the embryo and the zygote, the sex cells that have this sort of liberty to sort of grow into, you know, 1,000-fold bigger than what they were to begin with?
Yeah, I think, well, that's kind of like the idea of trying to understand what are the mechanisms behind that, right? And we'll have to like design specific experiments to figure out what are them. I'm not sure if I'm answering your question, but yeah, that's kind of like reading of it. Is that what you were asking?
Yeah, no, just how, like, maybe ahead of time think of how to uncouple those effects from the rejuvenation putative ones that you're observing.
Yeah, exactly. I feel like a lot of that is actually on the computational tools that we use today. Like, for example, we can identify a few transcription factors that produce a benefit that we want, right? And then it causes that effect, but it also causes a bunch of other effects that we don't want, right? Like the differentiation and other things like that. I feel like if we have more power for computational tools that can actually disentangle the mechanistic networks and we can find better targets or a better way to manipulate the biology, then we can get away with just doing what we need, right? Without the bad effects of using this.
That's about it, unfortunately.
Okay.
This is only the beginning, guys. You know, like if you're interested in this topic, A, talk to us, and B, we'll have working groups on this. Now, my question to you, what is
my question is, how can we create computational models that make use of biological data?
Okay, give me one.
Okay, go ahead.
Yeah, take us.
20 words.
How can we create a computational model that makes use of biological data in the same way the large language models do?
You just
Chat wants you to teach.